So today we are looking at disaccharides. So what are disaccharides? So disaccharides, as the name suggests, di always means we are talking about two things, right? So disaccharides, these are two monosaccharides which have been joined together, and then they end up to make what? What the result of that is what? Is a disaccharide. So a disaccharide is basically a situation where I've got two monosaccharide molecules joined together. Okay, so what happens here is, like we had discussed earlier on, the bond that's going to be formed between those monosaccharide molecules, between those two monosaccharide molecules, is going to be a glycosidic bond. So as you see later on, it's very, very important to be able to figure out exactly what kind of glycosidic bond do you have, because you can have a 1,4, you can have a 1,6 and a 1,2 glycosidic bond. And so they'll ask you questions such as what glycosidic bond is in sucrose, and you should be able to answer. So those are things that we'll be looking at. So like we said, this reaction that forms the disaccharides is a what? Is a condensation reaction. Why do we call it a condensation reaction? Because in the process, what happens when those two monosaccharide molecules come together, water is what? Is eliminated. So we call that condensation reaction, okay? Or a dehydration reaction. So what we are having here on top, you can see that equation, that's C6H12O6. Remember, we know those are monosaccharides, but remember, just from the molecular formula, we can't really be specific whether it's glucose, it's fructose, or it's galactose, because these are structural isomers, right? Meaning they've got the same molecular formula, but different structural formula, isn't it? So, but what we are aware of and what we are certain about is that these are monosaccharides because of that general equation. Okay, that we know. So we see there we've got two monosaccharides joining, and then what we are having is, so you see, how you can figure out the formula, the molecular formula that you're going to get for the disaccharide is basically add the carbon 6 and 6, you get 12, isn't it? Then for the hydrogen, add 12 and 12, you get what? 24. But remove two hydrogen atoms which are going to form what? Which are going to form water. So you see, instead of hydrogen being 24, it's 22, because two hydrogen atoms have gone to the what? To the formation of water. Then look at oxygen. Oxygen is 6 plus 6. So instead of being 12, you see that it's 11 on the disaccharide. Reason being because that oxygen has gone to you know that oxygen has gone into the formation of water. So yeah, so from that you don't have to memorize anything. So now what you're having here, you can see from those structures that what we have there is glucose. So can someone tell me that glucose, is it alpha glucose, is it alpha glucose or beta glucose, just from the structures that we have? Anyone that can help us? Is it, yes, is it alpha glucose or beta glucose? Because that's something that we discussed, and so that should be able to come out pretty quick from you. So in case you forgot, and then you know that you have to do some good revision, because this is something that we discussed just a couple of days ago. Anyway, so there, what you have there, you see you've got the glucose. I won't say whether it's alpha or beta, so that you can go and go through it. Okay, so now what I want to talk about is, you see the way we are numbering this glucose on the left, it's got numbers, right? Like we discussed. So what you're going to see there for those numbers is 1, 2, 3, 4, 5, and 6, right? 6 being there on the top carbon. Now, what is important in the naming of this bond? Remember, I mentioned that these bonds are named differently depending on where the hydroxyl groups or the OH groups, depending on which carbon they were attached to. So what you are seeing here is that you see on the first glucose, it's on what? It's on carbon 1. Whereas when you look at on the second glucose, it's on carbon what? Can someone answer that? What carbon is that hydroxide, the one that's being involved in the bond formation? What carbon is that hydroxyl group on? Hello? What carbon is it on? What carbon number is it on? Okay, so you people, you have to be revising, have to be studying. So you see it's on carbon what? It's on carbon 4. Since the numbering always starts on the far right and then you're going to your left. So that carbon there is what? Is carbon 4. So the on the glucose on the left, it's on carbon 1, whereas on the right, it's carbon 4. So we know that that bond is happening between carbon 1 and carbon 4. So take note of those numbers because we're going to use them in what? In naming our bond. Now, you see that now what has happened is the one hydroxyl and the hydrogen, they combine in a condensation reaction, of course, releasing what? Releasing water, which you can see there. And then you see that the way that bond has been written, that's a glycosidic bond. So that's a glycosidic bond. So the bond formed during the condensation reaction between monosaccharide molecules is called a glycosidic bond. This bond is formed between the carbonyl group of one monosaccharide molecule and the hydroxide group of another or group on another monosaccharide. So you see where that bond is going to be formed. So between the carbonyl group, so you can see everything be done there. So you see that bond is formed. Now a 1,4 glycosidic bond is from one carbon, oh sorry, is from one carbon 1 of one monosaccharide. Okay, let me just explain that. So when we say a 1 to 4 glycosidic bond, this means that this is a bond that involves uh, where the oxygen, where the bond is formed between carbon 1 of one monosaccharide and then carbon 4 of another monosaccharide. So if we have a 1 to 2 glycosidic bond, what does it mean? It means this is a bond between carbon 1 of one monosaccharide and carbon 2 of another monosaccharide. And there will also be 1 to 6 glycosidic bond, which again means it's carbon 1 on one monosaccharide and then carbon 6 on the other monosaccharide. So basically that's how you know. But for this for this maltose, it's 1,4 glycosidic bond. Okay, so the question that I was asking about the glucose, so it's alpha glucose. Yeah, so now that other one that we are looking at is done. Now let's now look at this other example. So what we had earlier on, that was glucose and glucose. So you're thinking, what's the name of that disaccharide? Anyone? Glucose and glucose. What is the name of that disaccharide formed? So
Maltose.
yeah, exactly. Thank you very much. So the one that was formed is what maltose. So why am I asking this? So this is what they will do in your test and everything. They'll just give you those things. So just draw that and then ask you to, yeah, sometimes because in biology you have quizzes. For quizzes, that's where you're allowed to write your answers, like where you can write something and everything. But for your tests, exams, it's just multiple choice. So in a quiz, that's where you'll be told to draw the structure of maltose. So yeah, you can be asked something like that. And that quiz actually also a quiz is just a university way of saying an exercise, right? So they don't say exercise, they just call it a quiz. Okay, so now here what we're then having is we've got alpha glucose. Why is it called alpha glucose? Anyone? And why are we calling it alpha galactose? And how could you tell the difference? So all those are things that we talked about last time, and it will be very good that you have these things on your fingertips, because that's how your questions come. So in case you are uncertain and clear about all these questions that I'm asking, take time to go through the previous videos. Anyway, so we are calling it alpha glucose because the hydroxyl group on carbon 1 is below the ring. Again, that's something that you can only understand if you check the previous videos in case you didn't go through. Anyway, so we've got alpha glucose and alpha galactose. Again, we see what is involved. It's those hydroxyl on carbon 1 and carbon 4. So just by looking at the carbon atoms involved, what bond will you call this? What will be the full name? So looking, because it's on carbon what? Carbon 1, the hydroxy on carbon 1 and the hydroxy on carbon 4. You already know this is going to be what? A 1 to 4 glycosidic bond, and there you have it. So lactose has got a 1 to 4 glycosidic bond as well. Now why is it important? Because sucrose doesn't have a 1 to 2 glycosidic bond, and that's something that they really like bringing up. So they'll just say which one of the following does not have a 1 to 4 glycosidic bond. That's how they ask questions. So in biology you really have to go into the details. That's why I even added that those questions for the exam, so that you can be able, when you study a topic, go through it, see which questions you can answer. When you fail to answer them, check through the notes, things like that. Yeah. Okay, so sucrose, what happens for sucrose is, like I've said, it's going to have a 1 to 2 glycosidic bond. So this means it's going to involve carbon 1 of a monosaccharide, and it's going to be bonding, or the interaction is going to be with carbon 2 of the other monosaccharide. So we know sucrose is going to be formed between what? Alpha glucose. There we've got beta fructose. So in order to make this work, since you see that number 2 there on the fructose is on the right side, so for the interaction to occur, as they are saying, there is a 180 and inverting itself. So what does 180 means? When you draw a straight line, a horizontal line, that's 180. So in short, what they're just basically saying, or if you draw a line which is vertical, that's still what? That's still 180. So it's more or less like you are looking at this that has flipped on itself. So like, it's like when you've got your hand, you've got your palm facing on the left, then the other aspect of your arm, of your hand, facing on the right. Then you just more or less just flip your hand so much that your palm is now facing you, or the palm is now facing the right. So it's just like you're turning your hand like this. So that's why you see that now the 2 which was on the right is now on the left, and therefore the interaction can occur. Okay, so you go on carbon 1, you see there's a hydroxide there. You go on carbon 2, there's a hydroxide there. And then when they come together, of course, you see water is formed as we would expect. And then you've got what? A 1 to 2 glycosidic bond. So those are things which are very important to take note of. Yeah. It's at this point that then we'd want to talk about starch. So, has everyone understood the concepts of glycosidic bonds, and also how the interaction happens for them to be formed?
Yes.
Okay, so now we'll talk about starch. So starch, as you know, okay. So starch, this is a polysaccharide. So we've moved from those disaccharides, and we're now talking about the what? We're now talking about polysaccharides. So starch has got two forms. So starch is a way of storing carbohydrates in what? In plants. Now we know that in human beings, this is stored in what? This is stored as glycogen, isn't it? So when we have excess glucose in the body, it's converted to glycogen. Now that's something that you learn in second year when you do your physiology and biochemistry. But in plants, there is no glycogen, it's starch, right? So starch has got two types. Okay, so the starch has got two types. There's the amylose and the amylopectin. Now it's important to understand the two as well as understand their differences. Because once you understand these things, you'll be able to understand how basically starch operates. So first things first, let's talk about the amylose aspect. So the amylose aspect, this is where you have the alpha glucose plus the alpha glucose combining. We know it's going to form maltose, but again you see it forms that. So we've got maltose 1 to 4 glycosidic bond, again another alpha glucose adds up, and then these just keep on adding up, adding up, adding up, adding up, until what's going to happen is that we're going to now have what? A structure such as that starch, a polysaccharide, which is more than 10, monosaccharide units. And then the hydrogen bonds, you remember the hydrogen bonds that we talked about when we first started? So those hydrogen bonds, they are going to be very, very important in the what? Stabilization of this structure. So you can see those hydrogen bonds interaction within. However, when you look at the shape, it's going to look like this shape that's on the right. So that's the shape that it's going to assume. Yeah. So okay, so starch is found in two forms, so I've already talked about. And then what's important about starch is that both forms of starch, they are formed from alpha glucose residues. Now, why is it important to bring that out? It's because when you talk about cellulose, cellulose is formed pretty much like starch, but the difference is that it's formed from beta glucose residues. Okay, so if you're still uncomfortable with the terms alpha, beta glucose, it's highly important that you check the previous videos. Okay, so I've already explained about the two forms. So the first form is amylose. That's what we looked at earlier on. So amylose is a coiled polymer or a helical shaped without branches. So when you look at a coiled polymer or a helical shape without branches, that's what I was showing you here. So you see, it looks like a helix or like a spring. Yeah, that's how you can think about like a spring, and then you see that it's got no branches in it. Yeah, so that's important to take note of about amylose. Then there's another form which is very branched, that's amylopectin. So let's first talk about amylose first. So amylose, the alpha glucose molecules are joined together by 1 to 4 glycosidic bonds, as we saw those bonds earlier on that I just showed you. Now, the angle that is formed between the carbon oxygen bonds, so when they basically say the carbon oxygen bonds, they're basically talking about, let me just slide there so you can be able to see. So they're basically talking about, you see that oxygen carbon bonds there, so that's just basically what they mean, huh? So I just want you to appreciate here, let me see if I can, yeah, so I just want you to appreciate that particular aspect there. Okay, I was trying to zoom to see if I can really bring it into focus, but anyway, so in short, they want us to, okay, yeah, that does it. Yeah, so basically it's that oxygen, the one that's forming the what? The one that's forming the glycosidic bond. And you can see that angle, the angle that is there is 109 degrees. So where else do you remember seeing an angle of 109 degrees? It's in the structure of water, isn't it? Yeah, it's in the structure of water. So you have to know these angles because you will be asked. So the angle formed between the COC bonds, carbon oxygen carbon bonds, between the rings is similar to the angle of water molecules, about 109 degrees. This angle means that the polymer is twisted into a helix. Again, that's something that I've explained, right? So it's like a spiral staircase. Or you can think of it like a spring. And you might remember that's how the DNA is, huh? But we'll discuss more on that when we start discussing the DNA. So the hydroxide on the carbon 2 of each glucose residue projects into the middle of the spiral. So what does it mean? As that spring thing is being formed, as that spiral shape is being formed, what happens is that the hydroxide group on carbon 2 of each glucose residue, that is to mean of every monosaccharide that is involved, projects, protrudes into what? Into the middle of the spiral. So it is now these hydroxide groups. So it's like a spiral is being formed. So it's like you're getting a spiral that's being formed, and then you're getting those hydroxide groups, which are actually going in the middle. So you see, there's a spiral shape, a helical shape formed. Then you've got on carbon 2, there are all those hydroxide groups from each glucose residue that's projecting in the middle. So now, because those are now in close proximity, and we've got the negatively charged oxygen and the positively charged hydrogen on those hydroxides, therefore, what you're going to get is hydrogen bonds are going to be formed between these what hydroxyl groups, which stabilizes the shape of the amylose polymer, holding it in its helical space shape rather. So remember what we talked about hydrogen bonds. These are weak, but when they are together, they can do a lot of wonderful things in that they can cause great stability, giving water its wonderful properties that it has. So hydrogen bonds are very important in that regard. Okay, so is everyone clear on amylose? How you can tear it apart from amylopectin? Okay, at least the different features of amylose, the bonds that it has, the angle that it has, how its helical shape is stabilized. What shape does amylose have? So if you can answer those questions, then you are in good shape. Amylopectin. So in amylopectin, it's a little bit more interesting because in amylopectin, the alpha glucose monomers are joined by 1 to 4 glycosidic bonds and 1 to 6 glycosidic bonds. So now this is interesting because you know that in the structure of glucose, you know that the carbon 6 is somewhere on top of the molecule, isn't it? So already the moment you hear that there's a 1 to 6 glycosidic bond, already you are thinking that you are going to have what? Some branching, isn't it? Already you are thinking that there's going to be some branching because when you check, look at where carbon 6 is. Let's see. Yeah, so look at where carbon 6 is. It's on top there, right? So I mean if you're going to have a bond 1,6 or 1 to 6 glycosidic bond, it means that it's going to be there and it's going to be up. So that's going to cause a what? A branching in your structure. So amylopectin has the 1 to 4 glycosidic bonds, which are just going to be normal, and then the 1 to 6 glycosidic bonds. And again, because of what I've just shown you and what I've explained, you will realize that anytime where you have that particular arrangement with regards to a 1 to 6 glycosidic bond, it's going to be a branched, okay? So it's going to be a branched point which is going to be introduced in the polymer. So that's an important point to take note of. So the branched point occurs every 24 to 30 residues, making a multi-branched compact polymer. So what they're basically saying is that you are going to have about 24 to 30 monomers, or the glucose residues, which are going to just be forming 1,4, 1,4, 1,4, 1,4 glycosidic bonds. And then on the 25th or the 31st, That's where now you are going to get what? That's why you are most likely to get so that's why you're most likely to get the 1 to 6 glycosidic bond, and so it's that 1,6 glycosidic bond that's going to lead to the branching. So that's why you're going to see a good degree of what, of branching. So that's something that's very very important to take note of. So yeah. Okay, so do you have any questions? Any questions? Okay, so something that I would like to make mention of. Okay, so something that I would like to make mention. Let's see if we can. Okay, so we've understood the branching and everything that occurs. Okay, so yeah. Okay, so that's basically so yeah. So both forms of starch are very compact. So what forms of starch are we talking about? Okay, let me just check if people are still in. So both forms of starch are very, very important. So that's to say, both amylose and amylopectin are compact. So what does compact mean? Remember, what are these polysaccharides used for? So these polysaccharides are going to be used for what? Are going to be used for storage, right? So if you want to form something, you'd prefer something that's compact, isn't it? Something compact is something that's closely put together. And so if you are packing, you're going on a trip, and then you just put your clothes uh, anyhow, you find that your suitcase may not contain as many clothes as it would contain if you placed the clothes compact, right? That's why if it will be different as though compared to if you put each cloth properly, neatly close together. You see that most clothes will actually go into your suitcase. So that's a key thing, because amylose and amylopectin are both important in the storage aspect in the plants. That's why these both forms of starch are very compact. Okay. However, amylose is what is coiled into a compact helical shaped, as we've seen, whereas amylopectin is branched, okay, to form a closely packed brush shape. So what you're going to see for amylopectin is you can see that shape of amylopectin. Okay, just pardon me. Yeah. So you can see that shape of amylopectin there. So you see here there is 1,4, 1,4 bonds. Again, by the 25th or the 31st, you have got that 1 to 6 glycosidic bond. And because of that, a branch has been created. So because of all these branches, what you're eventually going to see, it's that you're going to have those branches, and so that's important to take note of. So the function of starch is to act as energy storage molecule in plant cells, and the compact shape means that many glucose residues can be stored within a small volume of the cell. Okay, and this helps in saving space, as this particular plant is trying to save, to conserve and save these energy molecules, right? So that's something that's important to take note of. As well as saving space, converting the glucose molecules into polysaccharides reduces the number of molecules in the cells. Now this is very important because remember, water enters cells by osmosis. So if you had glucose molecules all over the place, that would affect osmosis, because that would mean that the cell will be very very highly concentrated. And remember that if the cell is highly concentrated, what is osmosis? Osmosis, water will move from where it's in abundance to an area which is very concentrated. So if glucose wasn't being converted to these storage forms, the cells would be so concentrated, the cells will have so much glucose, so much that water will be rushing and rushing and rushing into the cells. And then what happens? When the cells swell up, there is lysis, right? That happens. So the fact that these particular glucose molecules are going to be converted, okay, into starch means that the solution inside the cell is less concentrated, and so less water will enter the cell by what? Will enter the cell by osmosis. So that's something that's very, very important to take note of. So that's something that is important to take note of. So yeah. Okay, so storage polysaccharide, we already talked about that. So we've talked about starch, how it is formed. Now, such a compound, so starch is formed of an alpha glycosidic chain. So that's mostly what's going to be there, right? The 1,4 glycosidic bonds. And then such a compound where when you break it down, that's what hydrolysis means. It's just breaking something down using water. So when you break this down using water, and why do we use water? Remember, how were these things formed? They were formed by condensation reaction. Okay, and what happens in a condensation reaction? What happens in a condensation reaction? So what happens in a condensation reaction is that you're going to have water that's going to be lost, right? So if you want to break down these particular bonds, what do you have to do? You have to reintroduce the water that was lost. That way those bonds are going to be broken. So that's why we use water. So now if we use water and then we break down starch, the fact that it only produces glucose molecules on its breakdown, that's why it can be called glucan. So when you ask what's glucan, this is just basically starch. When it's broken down, you're only getting what? Glucose on hydrolysis. So this is very, very important because it's most important food source of carbohydrates. You can think of maize, you can think of cereals, potatoes, legumes, a lot of things. A lot of things where we definitely rely on this. So the two chief constituents of starch we've already talked about. It's amylose. Now amylose makes up about 15 to 20% of every starch. So for instance, if you've got starch, let's say you've got maybe 1 kg of starch, isn't it? 15 to 20% of that starch is going to be in the form of amylose, which is non-branching, a helical structure, right? So it's a helical structure with no branches. So 15 to 20% is actually going to be what? Is actually going to be to be amylose, whereas the remaining 80 to 85% is actually going to consist of amylopectin. Also amylopectin, which is going to have branched chains, which is 1 to 6 linkages at 24 to 30 glucose residues. So every time you need to know these percentages, amylopectin is more, okay, is more abundant than the amylose, and which makes sense because amylose is more compact. So if it's more compact, then you are thinking, because it's more compact, it doesn't take up much space. But because amylopectin is branched, it means that it's going to take up more space. So starch is a food reserve in plants and a major nutrient for animals. So it's a mixture of glucans. So we've already talked about what are glucans. Glucans is basically something where when you hydrolyze it, you break it down, you're just going to get glucose molecules as a result. So it's a mixture of glucans that plants synthesize as their principal food reserve. It's deposited in the cytoplasm of plant cells as insoluble granules. So when you look at a cell, you see those insoluble granules. And why are they insoluble granules? Because remember, we don't want the glucose to interact with the water. Because if starch was soluble, then we are defeating the purpose for which it was formed. Because the main reason why it's formed is we don't want it to be interacting with the water, with the fluids of the cell. Because if it's interacting, that will increase the concentration of the cell, and with the increasing of the concentration of the cell, that means that osmosis is going to occur, When osmosis occurs, what that is going to lead to is water entering the cell because it's too concentrated, and it can lead to bursting of the cell, which is cell lysis. So now let's now talk about glycogen. So starch, of course, that's how animals store their energy, but glycogen, that's how human beings store their energy. So, or animals in general, let's just leave it loose. So glycogen is formed from alpha glucose residues. So we know both starch and glycogen are formed from alpha glucose residues. Why do I want you to take note of this? Because we're going to talk about cellulose, and one interesting thing about cellulose is that it's not formed from from alpha glucose, right? It's formed from beta beta glucose. So that's why I want you to take note of all these things, because that's where your questions come from. So glycogen is a polymer of alpha glucose monomers in which the residues are joined by 1 to 4 glycosidic bonds and 1 to 6 glycosidic bonds. So just ending there, can someone tell me, do you think glycogen is branched or unbranched? Just by those statements about the bonds involved, the type of bonds involved, would you say glycogen is branched or it's unbranched? Okay, so the glycogen polymer is branched like amylopectin. So, by the branch points in glycogen occur every 8 to 12 residues. So, you see the difference, right? So, this is branched, glycogen is branched, but actually comparing it with, comparing it with the comparing it with amylopectin. So comparing it with amylopectin, you realize that this is actually what, even more branched. Okay, so glycogen is actually more branched than actually amylopectin, because remember amylopectin, we are saying it was having branches at about 24 to 30 residues, right? But with amylopectin, it's actually 8 to 12 residues, meaning that this is very, very branched, okay? And it's even more branched. Like, remember, we said amylopectin. We said amylopectin is brush-like, isn't it? But glycogen is actually more branched. So that's something important to take note of. Okay, and even though it's very much branched, but it's also compact. Because remember, the aim is what? The aim is to be compact so that you can store as much energy. So if it's not compact, then it won't really make sense that it's a storage molecule. So then this means that many glucose molecules can be stored in a single macromolecule, and it prevents them from being too high in concentration of glucose within cells. So that's something that I explained, so that cells don't get destroyed because of osmosis. Water moving into the cells causing the cells to burst. So the structures of starch and glycogen differ only slightly. Glycogen is greatly characterized by many side branches. We've already talked about that. Okay, then animal cells store extra carbohydrates as glycogen, which is sometimes called animal starch. So, because starch is something that was, maybe probably the first to be known and stuff like that. But so glycogen, because it's a storage form of carbohydrates, people loosely call it animal starch. Huh? After a human eats, the liver stores excess glucose or stores glucose as glycogen. Between meals, the liver releases the glucose to keep the blood concentration of glucose near the normal condition. So that's why when someone is fasting, or when you miss a meal, you don't just collapse there and then. The reason being is there is glycogen which was formed by your liver. So because that glycogen is there, that means that it's going to be broken down, and you still have energy. You still have energy. So that's something that's important. So that's why even if someone is fasting, they'll still be able to carry on with the fast because glycogen is being broken. So that's why someone just doesn't miss a meal and they die there and then. No, that doesn't happen. Okay, so we now look at cellulose, then we are done with the topic of carbohydrates. Then we are done. Then we just have to start lipids. So let's just do this so that we can finish off this topic. So again, one striking thing about cellulose is first things first, different from starch and glycogen is the fact that cellulose is made up of beta glucose as as the monomer, whereas all the others were having alpha glucose. So how do you know that it's beta glucose? On carbon 1, the hydroxide is going to be above the ring. In case that doesn't add up, check the previous videos where we explained those in full detail, but that's what beta glucose is. So you see what's happening there. You've got those bonds are formed, and then yeah. So one thing that's striking about this, you should be able to see those kinks which are being formed. We'll talk about them. So you see that kink there, that kink, that kink. We'll talk about them. Those are very, very important. Okay. So now let's explain cellulose. So there you see the structures. Okay. Let's go to our notes. Okay. Okay, lipids is there. Okay, let's see. Okay, great. Yeah, these are the notes I was looking for. Okay, great. Just here we do. So cellulose is formed from beta glucose monomers, right? That's something I've already talked about. So now these are joined by 1,4 glycosidic bonds. That's fine, that makes sense. But where the differences comes in is because it's beta glucose monomers which are involved. What happens is that in order to make that 1,4 glycosidic bonds, because these are because these are beta glucose molecules, every second beta glucose residue must be turned upside down for it to form the 1 to 4 glycosidic bonds. So what's going to happen is that as a result of this, because every second residue is upside down, every kink that bends the polymer one way is cancelled by the next kink which is in the opposite direction. So a kink is just something maybe like a bend or something like that, right? But because the fact that the second residue is going to be turned upside down, these kinks end up being what? End up being stretched. Because if this one was bent to the left, the other one will bend to the right. So you see that at the end of the day, you're going to have left, right, left, right, left, right, left, right. At the end of the day, when you look at the molecule, it's actually going to be straight. Okay, so that's why the polymer that you're going to get is straight, and because that is straight, that has got a lot of consequences, right? And one of them is that cellulose cannot be digested by the human body. But let's leave it there. We'll discuss that more. So as there are only 1 to 4 glycosidic bonds, there are no branches in the polymer. Yes, makes sense, because branches only exist when there are 1 to 6 glycosidic bonds involved. Again, these straight and branched polymers come together to form microfibrils. Okay, don't worry, I'll show you those. So basically what they're talking about, these microfibrils, these are more or less like small fibers, right? Which means that cellulose is strong. That's why even the human body can't digest it, and it's what we call dietary fiber. Okay, those things you say no, your body doesn't digest, but they're important because they prevent constipation. So that's a lot more of physiology, yeah. But you learn it, yeah. So the polymers making up the microfibrils are held together by hydrogen bonds between the what adjacent chains. So okay, let's see if I can find those pictures. Okay, yeah, okay, yeah. So you see those structures, right? So you see those structures are there, and you can see that you can see that those bonds, the oxygens are changing, right? You see that there on top it's up, like the oxygen that's joining the two glucose molecules. You see there it's on top, there it's down, there it's on top, there it's down. So that cancels any kinks which are there, and you can see the hydrogen bonds, which are interactions between the hydroxyl groups there. So because these bonds, hydrogen bonds, it's formed the microfibril, right? So it's very strong. It's very strong, and the tensile strength is what is very high. Huh? So that's something important to take note of. Yeah. So okay, we've talked about that. So the cellulose microfibrils are very strong when they're subjected to a pulling force along their strength, and this stress makes them an excellent building material. Okay, so important there. Humans found Humans have found many uses for cellulose, cotton fibers. So cotton is, and you just think of how difficult it is to cut a cotton fiber. So that's cellulose. We often wear cotton clothing, furniture, buildings are made from wood, which contain cellulose. Okay, a high percentage of that. So you see, just the fact that so you see, just the fact that it's just beta glucose and not alpha glucose, you see just the many differences which exist between starch, glycogen, and cellulose. So most animals, however, even humans are not able to digest cellulose because the enzymes that digest starch are unable to break the hydrogen bonds that hold the straight chains of cellulose. Even so, cellulose is a recommended part of our diet because it provides roughage or bulk that helps the body, maintain regularity of elimination. So that's just to prevent constipation. For humans, plants are a source of starch and fiber. Cows and sheep receive starch and nutrients from grass because they have special stomach where bacteria can digest cellulose. So cows are able to digest cellulose, and sheep, but not human beings. So basically, that's where the topic of carbohydrates comes to an end. So next time we meet, we start lipids. I would have started it now, but ending it on the middle and then starting it may be tricky. So in your free time, remember to go through it, and then yeah, that would help. So the next time we meet, we start the new topic. So let me just save